Hello and welcome to my channel. Today's topic is about the brainstem and spinal cord. That is to conclude the nervous system. So let's begin. The brainstem is a stalk-like structure, lies in the posterior cranial fossa and continues in the vertebral canal as the spinal cord, and composed of three major parts: the midbrain and the pons, or the bridge, and medulla oblongata. It's full of ascending and descending tracts, plus the nuclei for the cranial nerves, and its reticular formation contains centers for respiration, cardiovascular function, and regulation of consciousness. So it's an important structure, of course. This is a front view of the brainstem. As you can see here, this is the medulla oblongata, and this is the pons, and this is the midbrain here. The main features of the midbrain are the two cerebral peduncles, or the crus cerebri. Between them is the perforated substance of the brain. These two crus cerebri belong to the midbrain and constitute part of the tegmentum of the midbrain, and they are formed by descending fibers, mainly the corticospinal cortico-nuclear tracts. As you can see here, the oculomotor nerve is related to its lower border. Then comes the bridge, or the pons in French. The pons is simply a bridge between the two cerebellar hemispheres. This is called the basis pontis, with a pontine groove produced by the basilar artery at the front. And observe, please, the emergence of the trigeminal nerve from the anterior surface of the pons, with its medial root. The smaller root is medial, and which is the motor root, and the sensory root, which is bigger and lies lateral to the motor root. These constitute the trigeminal nerve. Now, at the lower border of the pons, near the midline, emerges the abducens nerve, and at this angle between it and the cerebellum, called the cerebellopontine angle, emerges the facial nerve and the vestibulocochlear nerve. That is nerve 7 and nerve 8. The brachium pontis, or the arms of the pons, are the middle cerebellar peduncles that connect to the cerebellar hemispheres. Lower down, we have the medulla oblongata. This constitutes the closed part of the medulla. Closed and open means this part is not a boundary of the fourth ventricle, but the upper part, which is here, but seen from behind, is a border or a boundary of the fourth ventricle. That's why it's called open. So here we have the closed medulla, and the upper part of the medulla oblongata is called the open medulla. Observe up the front the anterior median fissure that continues in the spinal cord, with obstructed in the middle here as the pyramidal tract, or the corticospinal tract, is crossing to the opposite side. This is called the pyramidal decussation. And then comes the anterolateral sulcus with the rootlets of the hypoglossal nerve emerging, and then the olive, which underneath has the inferior olivary nucleus, and behind the olive we have 9, 10, 11 nerves. That's glossopharyngeal, vagus, and cranial accessory nerves. The most lateral, this part, is called the inferior cerebellar peduncle. So we have the middle cerebellar peduncle, which is the brachium pontus, the inferior cerebellar peduncle coming from the medulla oblongata, and the superior cerebellar peduncle, you can see it at the back of the midbrain. The posterior surface of the brainstem, this is the posterior surface of the thalamus with the pulvinar projecting. You can see the tectum of the midbrain and the pineal gland. This is the pineal gland. This is the cavity of the third ventricle. On the tectum of the midbrain, you will find the superior colliculi and the inferior colliculi, called together the corpora quadrigemina, with each projecting into a brachium. The brachium of the inferior colliculus connects it with the medial geniculate body, and the brachium of the superior colliculus connects it with the lateral geniculate body. The superior colliculus is part of the visual pathway, while the inferior colliculus is part of the auditory pathway. Now, this is the superior cerebellar peduncle I was talking about. You see that with the superior medullary velum in between, that constitutes the roof of the fourth ventricle. This is the fourth ventricle. This is the back of the pons with the middle cerebellar peduncles. The floor of the fourth ventricle, the upper half, is made by the pons, and you can see a medial eminence here, and facial colliculus produced by the facial nerve, nucleus of the facial nerve. Then comes the lower part of the fourth ventricle with the hypoglossal triangle and the vagal triangles in the floor. You can see the inferior cerebellar peduncles projecting from the medulla oblongata to the midbrain. And if you look closer here, you see longitudinal elevations. This is the graceal tract, and lateral to it you have the cuneate tract, both ending in tubercles, the graceal ending in graceal tubercle, and the cuneate ends in the cuneate tubercle. They carry proprioception and fine touch sensation from the body. The graceal carries it from the lower part of the body, and the cuneate carries it from the upper part of the body. The medulla oblongata continues in the vertebral canal as the spinal cord. That's why the spinal cord is called the medulla spinalis. The spinal cord is in the vertebral canal and extends down till the level of between L1 and L2 vertebrae, and the end of which is called the conus medullaris. See, this pointed projection, pointed structure here is the end of the spinal cord, is called the conus medullaris, and it has cervical and lumbar enlargements to give way for the brachial plexus and the lumbar plexus, lumbosacral plexus. And it's divided into segments. Each segment gives a pair of spinal nerves. The spinal nerves arise by rootlets, as you can see here, that merge into a single root, and then the posterior root and the anterior root unite at the intervertebral foramen and exit as the spinal nerve, which after exiting the intervertebral foramen, it divides into ventral ramus and posterior ramus. So don't confuse rami with the roots. All the nerves pass through the intervertebral foramina except C1, which passes above the C1 vertebra. You all know that we have 8 cervical nerves and 7 cervical vertebrae. As you go down, the rootlets or the roots of the nerves become longer because the faster growth of the vertebral column than the spinal cord. it pulls down the roots of the spinal nerves with it and forms a structure like that looks like the tail of a horse. It's called the coda equina. It's composed of the roots of the lumbosacral plexus. Finally, C1 through C7 are above its corresponding vertebrae, C8 below C7, and the rest are of the nerves lower down, going down, are below their corresponding vertebrae. This is a picture to show you a real picture of the coda equina, and observe please that the roots becomes longer as you go down to finally form the coda equina, and this is the end of the spinal cord, the conus medullaris. In this picture you can see the cervical enlargement and the lumbar enlargement. The cervical enlargement that gives off the brachial plexus, so you need more space for more cells. That's why it's larger than the rest of the spinal cord. And the lumbar enlargement for the same purpose is to give space for the lumbosacral plexus. Relation of segments with the vertebrae. The segment gives rise to rootlets, then merge into roots, then the nerve gives off rami. Don't confuse these rami with other rami given by autonomic nerve, which is the sympathetic precursors of the sympathetic chain. The white rami myelinated fibers and gray rami unmyelinated fibers. The lower motor neurons lie in the anterior horn of the spinal cord, of the gray matter of the spinal cord, and each segment you can detect the level of, for example, spinal cord injury by examining the piece of skin supplied by a certain segment. This is called dermatomes. It means the sensory map on the surface of the skin. It helps to determine the level of the injury if you suspect an injury in the spinal cord. The term myotome applies to muscle fibers supplied by a single segment, and in this segment there is what's so-called the reflex arc, composed of afferent sensory fibers to the dorsal root ganglia of the pseudounipolar cells in the dorsal root ganglia, the axons of which goes to the interneurons in the gray matter of the spinal cord, and then these interneurons project to efferent fibers through the same nerve back to where it came from. This is called the reflex arc. The arc goes this way to the dorsal root, relays on the interneurons in the middle, and then gives off the ventral root of the arc that goes with the same nerve back to its source. This is a map of skin map of the different spinal cord segments. You should be knowing that you should be knowing this map to help you determine the level of the injury in the spinal cord, as you can see here. This is another map, but not with numbers, segment levels, but with the name of the cutaneous nerves supplying the corresponding areas. You can study that in details in the corresponding lectures that belongs to the upper limb and lower limb, for example. This is another map of the dermatomes of the lower limb. We have what's so-called referred pain along also spinal cord segments, but because of the plexus is formed So there is exchange of information between different levels of the spinal cord. The pain doesn't have to be over the organ the piece of skin lies upon. For example, you can see how the liver pain is related near the midline, how the kidney pain is referred along with the spinal nerves to the suprapubic region, and etc. This is a map of the different parts or different areas for referred pain all over the body. Now, it was mentioned that the spinal nerves emerge from the intervertebral foramina. They give rise to ventral rami and dorsal rami. The dorsal rami never form plexuses, means they don't exchange information, they don't intermingle with each other. The ventral rami, on the other hand, these form plexuses. We have the cervical plexus, for example, as you can see here. This is the brachial plexus. We have the lumbosacral plexus lower down. This is the lumbosacral plexus. So these plexuses ensure that there is harmony in the movement of the muscles, and to save place, to save space, without the plexuses, the diameter of the spinal cord will be huge because of the cells supplying the muscles. So the plexuses save the space in the spinal cord, as well as achieving the harmony and the actions of the corresponding muscles. This is also a picture of the lumbosacral plexus in situ. As the spinal cord is a continuation of the brainstem, so it takes the coverings of the brain with it. It's covered also by the dura and arachnoid and pia mater. All these pass through the foramen magnum. The dura mater is the one you see here. It's open and reflected. It's one layer, and it's attached to the margins of the foramen magnum and to the intervertebral foramina at the site of the exit of the nerve. Actually, it encloses the beginning of the nerve and becomes the epineurium of the nerve later. The epidural space means the space between the dura and the vertebra. It's filled with fat and also a series of veins called the vertebral venous plexus. The dura fuses with the arachnoid at the level of S2, as you can see here. The pia mater continues and pierces the dura, and the arachnoid continues at the outside as the filum terminale externum. We'll talk about this in a while. The arachnoid goes with the continuation of the dura, and it's adherent to it, and sends sleeves around the exit of the spinal nerves. It's prominent here. You see the sleeves, it gives the dura giving with the exit of the spinal nerves. Don't confuse it with these projections. These projections come from the pia mater, called the denticulate ligament, between the nerves. But I'm talking about the sleeve around the nerve. Okay? And the subarachnoid space also continues with that of the cranial cavity, so it contains CSF. Since the spinal cord ends at the level of L2, so between it and S2, the site of fusion of the arachnoid together, there is a space filled with the cauda equina floating in CSF, collection of CSF. This is called the lumbar cistern, which extends from L2 to the level of S2. The lumbar cistern is the site for the lumbar puncture. You will be using it to introduce anesthesia, for example, in pregnant women giving labor, for example, or to take a sample of the CSF, or to introduce different kinds of drugs, or to introduce a dye and take a picture. This is called myelogram. This is a myelogram of the spinal cord. And now you can see these projections. Now remember, you are in the CSF cavity. These projections look like the projections produced by the PEA, but the projections produced by the PEA should be between these projections. These are the sleeves I am talking about, the sleeves that continues around the beginning of the spinal nerve filled with CSF in this case. The PEA matter forms the denticulate ligament, this dentate-like ligament going laterally attaches to the inside of the dura, and lower down it becomes like a rope, forming what's so-called the filum terminale. This is the filum terminale. This is not a nerve root. It's simply the continuation of the PEA. The filum terminale has part inside the lumbar cistern called the filum terminale internum, and then after piercing the arachnoid and dura, it's called the filum terminale externum, to be attached to the back of the coccyx to fix the spinal cord. The spinal cord is kept in place due to the dural attachments to foramen magnum and the intervertebral foramina, also to the denticulate ligament and the filum terminale and the exits of the spinal nerves. All these factors are responsible for fixing the spinal cord in place. Now, if you take a look at the internal structure of the spinal cord, with this type of stain, the gray matter appears light and the white matter appears dark. So this is the white matter of the spinal cord, and this is the gray matter in the middle. Opposite that, the arrangement that's in the brain. It's roughly H-shaped and has posterior horn and a ventral horn or anterior horn, and in the thoracic and upper two lumbar regions, it has an extralateral horn. This extralateral horn you can find at thoracic regions from T1 down to L2 and S2, 3, 4 segments, and these responsible for giving the origin of the autonomic nervous system. In the thoracic region being responsible to give the origin of the sympathetic system. In the sacral region, in the S2, 3, 4, gives the origin of the pelvic splanchnic nerves, or the S2, 3, 4 nerves that carry parasympathetic supply to the different organs of the pelvis. You can differentiate between the different levels of the spinal cord according to the amount of the white matter in ratio to the gray matter. As you go lower down, this is the sacral region. See, the gray matter is ratio to the white matter is much bigger than that of the cervical region, for example. And the shape of the gray matter in the middle. It's roughly an exact H in the thoracic region. This is the lumbar region where the gray matter, the posterior horn and anterior horn, are rather thick. And in the cervical region, the abundance of white matter in the cervical region, because all dendrites now, all tracts now are collected from the body and go up to the brainstem. And also, there is a phenomenon called shelving. You can find a shelf-like formation of the anterior horn of the gray matter of the spinal cord, as you can see here. So this is cervical, this is thoracic, this is lumbar, and this is sacral segments of the spinal cord. Now, the gray matter, when studied, it was found that it's made of multiple nuclei, means collections of gray matter with different functions. These are called the lamina of Rixod and constitute 10 areas, starting for lamina 1 at the posterior tip of the posterior horn till lamina 10 at the middle around the central canal of the spinal cord. And they contain different nuclei, means collection of cells that serve a certain purpose and give rise to certain tracts in the spinal cord. These are called the lamina of Rixod. The anterior horn cells, the anterior horn is motor, the posterior horn is sensory, and the intermediate horn is autonomic. Types of the cells in the anterior horn, motor neurons are lower, they are called the lower motor neurons, and they are of two types mainly: the alpha motor neurons that supply the skeletal muscle fibers itself, and the gamma motor neurons that supply the muscle spindle within the muscle. These are called the intrafusal muscle fibers of the muscle spindle. And each constitute a motor unit. The lower motor neuron with the nerve supplying a series of muscle fibers. This is called the motor unit. Posterior horn cell groups, as seen here, divided into several nuclei according to Rixod, and each receive special type of sensation. Pain and temperature, for example, being at the very tip of the posterior horn. It's called the substantia gelatinosa of Rolando, as you can see here, receiving the Lissauer's tract. Lateral horn of the spinal cord contains intermediolateral and intermedial cell groups, and they are autonomic cells, as mentioned before. In the thoracolumbar region, they give rise to the preganglionic sympathetic outflow. In the sacral region, they give rise to preganglionic parasympathetic outflow. The white matter of the spinal cord is divided into anterior funiculus, lateral funiculus, and posterior funiculus. Funiculus means big fascicle. It's a collection of nerve fibers and tracts, long tracts. We have a series of short tracts and series of long tracts. The long tracts comprise almost the most of the white matter, the ones you see here, but I'm talking now about the short tracts of the spinal cord, example of which are the septomarginal tract, the comma-shaped fasciculus between the gracile and cuneate tracts, and the tract that surrounds the whole of the spinal cord and connects different segments together. This is called the fasciculus proprius. You can divide it into anterior part, lateral part, and posterior part of the fasciculus proprius. These are the fasciculus proprius, the septomarginal, the comma-shaped are examples of short tracts that stretch through a few segments of the spinal cord, connecting them together. Now the long tract, you can divide them into sensory tracts and motor tracts, and this is the map of the motor and sensory tracts, sensory being in blue and motor being in red. You can divide the motor into pyramidal tracts and extrapyramidal The most prominent, of course, of the pyramidal tracts are the lateral corticospinal tract. The extrapyramidal tract means that these tracts mostly do not reach the cortex, but different areas, lower levels than the cortex, like originate from a red nucleus, is called the red rubrospinal, from the reticular formation, reticulospinal, from the olivospinal, from the vestibular nuclei called vestibulospinal. All these aid in the adjusting or the accuracy of the movement of the body. If you go, on the other hand, to the sensory section, dorsally is occupied by the gracile and cuneate, which carry fine touch and proprioception from all over the body, and you have different ascending tracts carrying proprioception and extra information to various parts of the brain, like the anterior and posterior spinocerebellar tracts, for example, the spinothalamic tracts that carry pain and temperature from all over the body. So these are examples of the long tracts of the spinal cord. Vascular supply of the spinal cord. It's a long structure, so it takes from nearby arteries, but the major blood supply comes from posterior spinal arteries. There are two arteries from the vertebral artery as it goes up in the foramen magnum. Anterior spinal artery, which is single, formed by the union of two anterior spinal arteries coming off the vertebral arteries as it goes up as well, entering the skull. And the branches of these spinal arteries, as they go inside the spinal cord along the spinal nerves, they are called radicular arteries. The venous drainage is through a series of longitudinal channels that connect with anterior and posterior spinal veins, and these are connected to the internal vertebral venous plexus inside the vertebral canal, and which connects also to the external vertebral venous plexus outside the vertebrae. They are all interconnected, so they are channels for spreading of metastasis from different tumors, especially on the posterior abdominal wall, like the kidney, for example. This is a picture of the internal vertebral venous plexus connecting to the external vertebral venous plexus on the outside of the vertebrae. I hope you got the maximum benefit from this lecture, and thank you very much for watching.